Longevity is not the result of a single molecule, gene, or nutrient, but emerges from a complex interplay of biological networks that maintain cellular integrity over time. Among these, autophagy, a process that recycles damaged cellular components, plays a central role in protecting cells from the accumulation of metabolic waste and stress-induced damage. Increasingly, researchers are recognizing that specific vitamins act not just as nutritional cofactors, but as regulators of gene expression, signaling pathways, and enzymatic functions that directly govern autophagic activity. Through a systems biology lens, we can begin to appreciate how these vitamins interface with energy-sensing pathways, inflammation modulators, redox balance, and mitochondrial health to support longevity at every level of cellular function. Take, for example, vitamin B3, niacin, a precursor to nicotinamide adenine dinucleotide, NAD, which functions as a central molecule in cellular energy metabolism and redox reactions. NAD levels naturally decline with age, and this drop is tightly linked to reduced autophagic efficiency and impaired mitochondrial quality control. Through its influence on sirtuin enzymes, especially SIRT1 and AD, plays a critical role in turning on genes that initiate autophagy and suppress inflammation. These sirtuins also interact with a FOXO family of transcription factors, which are known to extend lifespan in multiple species by regulating stress response and autophagy genes. By supporting NAD availability, vitamin B3 enhances this regulatory axis and indirectly preserves genomic stability and mitochondrial function. Vitamin D, another key player in this network, is increasingly appreciated not just for its role in bone and immune health, but for its genetic influence on autophagy-related pathways. It acts as a transcriptional regulator by binding to the vitamin D receptor, VDR, a nuclear receptor that modulates hundreds of genes. Among these are genes involved in autophagosome formation, lysosomal acidification, and inflammatory cytokine suppression. Chronic vitamin D deficiency is associated with impaired autophagic flux, increased oxidative stress, and the progression of degenerative diseases, all of which undermine longevity. When viewed as a gene-level switch, vitamin D becomes less of a passive nutrient and more of a master regulator of cellular cleanup and renewal. Vitamin B2, riboflavin, and B6, pyridoxine, contribute in more biochemical ways, serving as cofactors for key enzymes in energy production and amino acid metabolism that fuel the autophagic process. Riboflavin is required for flavoproteins involved in mitochondrial respiration, ensuring that ADP production is sufficient to support the energetically demanding phases of autophagy. Vitamin B6, on the other hand, facilitates the synthesis of neurotransmitters and glutathione, one of the body's most potent antioxidants, helping to maintain redox balance, a prerequisite for proper autophagy initiation. Deficiencies in these vitamins may not cause immediate symptoms, but they can impair autophagy at a foundational level by weakening the metabolic context in which it occurs. Vitamin C and E, as antioxidant vitamins, have a more nuanced relationship with autophagy. On one hand, excessive oxidative stress is a major trigger for autophagy. On the other, chronic oxidative stress that goes unmitigated can impair lysosomal function and damage autophagic membranes. These vitamins help create an optimal oxidative environment, sufficient stress to induce autophagy, but not so much that it overwhelms the system. Vitamin C also plays a role in iron metabolism, which affects lysosomal health and autophagosome maturation, while vitamin E protects membrane structures, including those forming during autophagy, from lipid peroxidation. In this way, antioxidant vitamins modulate the balance between autophagy stimulation and cellular protection. What systems biology reveals is that these vitamins do not work in isolation. Their effects converge on key regulatory nodes such as mTOR, AMPK, and sirtuins, major hubs that sense nutrient availability and determine whether the cell will prioritize growth or repair. For instance, vitamin-dependent modulation of NAD and ADP levels feeds into AMPK activation, tipping the balance in favor of autophagy. Meanwhile, suppression of mTOR activity, essential for autophagy initiation, can be indirectly achieved through the metabolic adjustments brought about by optimized B vitamin and D status. Each vitamin nudges the system toward a homeostatic state where cellular maintenance is prioritized, especially under conditions of mild stress, such as calorie restriction or intermittent fasting. Additionally, The gut microbiome plays a supportive role in this axis, both producing and modulating the absorption of several B vitamins. A disrupted microbiome can impair vitamin synthesis and uptake, thereby weakening autophagic tone and accelerating systemic inflammation. This suggests that any systems-level approach to longevity must also include gut health as a mediator of vitamin autophagy dynamics. The real strength of the vitamin autophagy longevity axis lies in its integrative capacity. Instead of forcing autophagy through extreme interventions like prolonged fasting or pharmacological agents, this approach supports the body's natural regenerative rhythms through subtle, sustained optimization of nutrient availability. It empowers each cell with the tools it needs to maintain homeostasis, adapt to stress, and extend its functional lifespan. In conclusion, systems biology illuminates the elegant complexity of how vitamins help regulate autophagy and influence longevity. These nutrients do far more than fill metabolic gaps. They serve as regulators, modulators, and stabilizers of the very pathways that determine how cells age and repair. By optimizing vitamin status with the same precision we might apply to fasting windows or macronutrient ratios, we unlock a deeper, more holistic approach to anti-aging, one that works not by forcing cells to survive, but by giving them the information they need to thrive.